analog. Are you confused already? Yes. Raise your hand. Okay, Donna, help them out.
All right. Hi. Good afternoon. I'm Donna Viola again. My presentation is Life on Mars, Endolithic Biodiversity of a Terrestrial Analog. So before I get going, just a couple of quick acknowledgements. My INDS faculty advisors, Ian George is not here, and Dr. Freeland, who we just tried to call on Skype, and unfortunately Skype's not working. And also my INDS advisor, Patricia Lanu. You guys have put up with all my crazy ideas over all these years, so I appreciate that. I'd also like to thank Hal Schreier with Marine Biotechnology and UMBC Biology for lab work assistance. He's been instrumental in getting materials, working with some of my protocols, processing my samples. I'd also like to thank David Eisenman, who provided me with lab space, and also people in his lab for day-to-day laboratory assistance. Sherry Petty from the UMBC Keith R. Porter Imaging Facility for assistance with my microscopy work. Jocelyn Ruggiero from Johns Hopkins University for helping me clarify my research topic and help with some original references. Heidi Beamer, Kevin Newman, Andy Gilkey, Max Fagan, and Sigrid Arnjad for helping me out in my field work, and also funding from the Maryland Space Grant Consortium. So,
and we won't count that as a part of your time.
Yes. That out of the way. I went to Mars in February. Not quite this Mars, 100,000 kilometers from Earth, with an average surface temperature of minus 60 degrees Celsius and a surface pressure that would boil your blood away if you were to step outside without a spacesuit. I went here to the Mars Desert Research Station in southeastern Utah. This is an analog station operated by the Mars Society, and it's staffed by volunteer crews. So for 2 weeks, different groups of volunteers go out here, and you live and work as if you're actually on Mars. I was a part of Crew 99, commander with 5 other students that I met in my summer program last year. Each one of us had our own scientific objectives and also responsibilities to keep the habitat operational and keep the simulation running smoothly. My particular project was looking at endolithic microorganisms. And what these are is basically microbes that live inside of rocks. And you can actually see a green layer. I'm going to send this around. But just below the surface, you can see a green layer. And that's actually cyanobacteria that live inside of this habitat. And so I'm looking at this as a model for potential life on Mars. So we know the surface of Mars is not that friendly, even if you're a person or a microbe. It's not a good place to be. But when you move inside of these samples, inside of the rocks, the environment becomes a little bit more hospitable towards life. So I wanted to look at these really as a potential model for life on Mars. So the purpose of my project was to look at the biodiversity of endolithic organisms in this particular Mars analog environment. And to look at the distribution of these species across their spatial locations as well as the rock types in which they're inhabiting. And I also wanted to use this information as well as what we already know about the environment on Mars and try to extrapolate this for potential endolithic habitats that we might be able to find on Mars. And this required a very interdisciplinary approach, looking at biology, geology, geographic information systems, and planetary science. So in methodology, I took this from several different approaches. So sample collection was my top priority. That was kind of what I was doing when I was out in the field. And so this was, each sample location was chosen based on evidence that there was water at some point in the past in this environment. So dry stream beds, gypsum deposits, these kinds of things. I collected samples. So at each site, I collected GPS coordinates to get a spatial distribution of where these samples actually came from. And I also just did some visual geologic classification of both the rock type itself, of each sample, as well as the surrounding area and what kinds of processes had formed those environments. Brought the samples back to UMBC, so some of my lab work. Fluorescent microscopy was used to confirm that the green layers in these rock samples are actually microbes and not some sort of mineral deposits inside of the rocks. And I was also doing some DNA extraction on these, so scraping off these green layers using a soil DNA extraction kit to pull the DNA out. Then used PCR to amplify the 16S rRNA genes. So what these are is very good for species classification. Every bacterial species has its own unique 16S sequence, so it's really good to distinguish between the different species. And so that was used to just get the community DNA from each sample. And then to separate these into their actual species, we're using a denaturing gradient gel electrophoresis, that's this DGGE. And it basically will separate the 16S sequences in the community DNA based on their species, based on the charge and the size of that sequence. And that's useful for getting the species distribution and kind of figuring out what's actually present in those samples. So using these plus some local geologic maps, I was trying to determine the distribution of these biological species relative to their spatial locations, as well as to the rock types where they were found. And then use that to compare to Mars and extrapolate these potential habitats on Mars. So for my integration strategy, the primary technique I was using was constructing a comprehensive perspective. So using both the biology and geology, looking at these habitats and trying to understand it from both perspectives to get a great understanding of what's actually going on in these environments. And I also used the method of extension, so taking the concept of terrestrial habitability and what we know about life here on Earth and trying to extrapolate that and apply it to the potential habitability of Mars. And some key points of common ground that I came across is that these endolith specimens, they have both biological and geological properties inherent in them. And also using terrestrial analogs as a sort of common ground between Earth and Mars. So we can't actually go to Mars right now, but we can use these kinds of environments to simulate the kinds of things that are going on our neighboring planet. So this is just a quick look at my sample sites. So this red one right here, that was the habitat itself. And then I had 7 different sample sites kind of spread across the area. The distance here is about 5 kilometers, so they were all fairly close to the habitat. So these are some of my results. So this is looking at fluorescent microscopy, and this is looking at these rock samples under 3 different kinds of fluorescent wavelengths and looking at the returns on that. So this was 2 control regions that you have. This is both uncolonized regions, so there's no evidence of any kind of bacterial colonization in these. And you have an inside and outside face of the rock. And what the fluorescence is really showing is that you kind of see the same patterns in all 3 different types of fluorescence, lighting up in the same places. Compare that to 2 colonized regions that I looked at. You can see that there's some repetition of the pattern across these, but there's a lot stronger returns under green fluorescent light, and that emits as red. So that's this bottom left here. And you can see these more brought out patterns. And I saw the exact same pattern looking at just cyanobacteria suspended in water. They only lit up under this green fluorescence. So I basically took this as an indication that yes, the green stuff inside of these rocks is some kind of organism. Of course, throughout the process, as science usually is, I ran into a few challenges trying to get the DNA out of these. So in a few weeks, tried to pull this out, tried looking at the samples using a spectrophotometer. It was below the detection limit, so there did not appear to be any DNA in those samples. I tried amplifying the 16S RNA genes, ran them in a gel to see if I can get any product out of it, and there's no product whatsoever in here, not even for a positive control. So I put even just pure E. coli, and I couldn't get any DNA out of it. So clearly something's wrong with the procedure that I'm using, or something in the lab that I'm using. So what we did, Dr. Schreier took some of my samples to his lab in Baltimore, did the same thing, ran it through a gel, and you can see all these bands right here. That's DNA. There was DNA in these samples all along. So we're not quite sure what the problem was in the lab here, but you can see no DNA here. This was a no DNA control, and everything else had DNA out of it. So heartened by this result, we continued on with the procedure, and this is what the results of my denaturing gradient gel electrophoresis look like. So like I said, what this is doing is taking those community 16S RNA sequences and separating them based on their size and their charge. So each one of these bands in here represents a different species. And this is fairly new data. I just got it middle of last week. But what this shows is that you have a lot of different species inside of each one of these samples. So some of my preliminary conclusions at this point is that we have these complex communities inside of these endolithic habitats in an environment that doesn't seem to have any life in it at all. And so, yeah. And then, okay, so I'm also using this to say, kind of projecting, that we usually find endolith colonies in these dry, arid, sedimentary type environments here on Earth. So what I'm positing is that when we're looking for these kinds of organisms on Mars, that we should look in these sorts of environments that have been formed by some kind of water-related processes. So we see gullies, we see channels on Mars, and these would be a good target to look for these kinds of organisms. Work's not over yet. I have extracted DNA from some additional samples that I had. We're going to be running another denaturing gradient gel electrophoresis this week. Once I have that, I'll be able to really look at these relative to the different locations and the rock types. So actually that's what I've had to run before to try and get my samples to Baltimore so we can test them. If I had another month or two, I would also like to do some sequencing. So actually, take those sequences, figure out what those species are, and see how they're distributed across space and time. Fortunately, graduating in a couple of weeks, so we don't have enough time for that. Of course, in the very distant future, I would like to actually go to Mars and look for these kinds of organisms myself. Thank you. I will take any questions.
I have a very important question. In the first photo of the crew, was that Nutella in the photo? Did you guys, is the crew allowed to eat Nutella?
It was World Nutella Day.
Okay.
February 3rd or something? I forget.
I didn't know.
But we looked it up, we read online that it was World Nutella Day, and we happen to have Nutella. It's something that's kind of shelf stable, you can have that. And so we celebrated.
And a follow-up question, could you explain what PCR is?
Sure. So PCR, polymerase chain reaction. So what that's used for is basically when you have a little bit of DNA in a sample, it basically takes that and it multiplies it over and over and over again. And so you have, starting with a little bit, right, you can have just a couple of bits of DNA in there, and then you use PCR to produce more of it, and that way you have enough that you can actually analyze.
So you handed around one rock sample. How many different rock types did you examine to try to find these? And did they all seem to show the kind of evidence that the one you showed us had? Or not?
So they all had, you could see the green content inside of all of them. They were mostly sandstone and conglomerate type samples, just based on the environment. That's kind of what it's mostly made of. And yes, I'm hoping to be able to actually compare the species and see what those distributions look like. I have a couple more samples to process though, so I don't quite know the biology in it.
So you said that your hypothesis was that if this information was taken to Mars, it would impact where you would look for endolithic organisms in areas that had previously had water. Does that mean that you compared samples that you gathered from areas that had previously had water with areas that did not have water? Or were you only looking at areas that had at some point in time had water?
Well, the entire environment itself was really formed underwater. So there were a lot of channels there, but most of it was deposited in a sea environment. So really everything in that area had water at one point. But yeah.
Well, Donna, I was grateful to have the opportunity to touch base with you while you were in this zone of, oh my goodness, this is not working out. That's what I remember. She kept her chin up, but when she'd say, well, I have to go back and check the lab, maybe something's happened while I've been over here. Mm-hmm. So it's Dr. Eisenman's equipment? Is that what's the bottom line with the lab here on campus?
So it seems like the particular program I was running on his PCR machine wasn't working right for some reason. We haven't had a chance really to explore that too much. I just realized that we could get it to work on Dr. Shire's equipment in Baltimore.
So if there weren't a Dr. Shire and his equipment to use,
yeah, we wouldn't have really had anything. I mean, the fact that we didn't get any positive controls working was kind of a big heads up that something was wrong. Because even if there was no DNA in those samples, there should have been some kind of product coming out of the positive controls. But it was trying to figure out what the problem actually was. That was frustrating and enlightening at the same time.
well, frustrating and enlightening. I'm sure you followed what was going on or supposed to go on this weekend that didn't in terms of Florida. Do you have any comment for the rest of us since you know how to analyze that better than we?
I mean, well, that with the shuttle launch is just technical difficulties.
Yeah.
That they're working through and we'll hopefully be able to launch.
Well, hopefully
soon.
when you're on your way to Mars, there won't be technical difficulties.
There are always technical difficulties, but we will figure it out and work through
It is my great pleasure to give you this cord. Donna, as you might have expected, is also a student graduating with Interdisciplinary Studies honors. She's graduating summa cum laude. I think she has one B on her transcript someplace.
Two Bs.
Two Bs someplace on her transcript. And she will be going off in the fall to the University of Arizona to work on her PhD. Maybe this summer you'll get to finish that in a track in astrobiology.
Planetary sciences with a minor in
Planetary science.
astrobiology.
Okay,
Yes.
that's it, planetary sciences. So we wish you well and thank you for your presentation.